In this video, we'll be reviewing my favorite kind of macromolecules, proteins. Proteins make up more than 50% of the dry mass in our cells, serving as important macromolecules for chemical reactions, serving structure and integrity for our cells, as well as some extremely important biological and chemical reactions and interactions. I would argue the most important type of proteins are enzymes, right? Enzymes essentially are these proteins that speed up chemical reactions. They maintain and control the metabolism in our cells, but also speed up chemical reactions by acting as catalysts or agents that selectively speed certain reactions. For example, the enzyme sucrase accelerates sucrose into glucose and fructose, right? They break apart certain substrates and produces different individual monomers, right? And that's what enzymes do. Now, the monomers of proteins, the monomers, are known as amino acids. Amino acids. There are only 20 of them, right? You can practically memorize all the amino acids if you want. There are only 20 of them. Now, you might be like, okay, there are tens and thousands of different proteins in the human body. How are they only constructed of 20? Well, that's because each protein is different in length and different in structure and different in the types of bonds they create, which causes them to be diverse. A single change in a bond can, cause a whole different protein. So that's why even though this number amino acids is quite small, the combination of different bonding arrangements make tens and thousands of different types of proteins. So then the polymer of proteins are known as polypeptides. Polypeptides, right? And they're an arrangement of 20 different amino acids just sequenced differently. They can go on hundreds and thousands of different sequences to then make a protein. Let's go over the structure of amino acids, right? Amino acids. Amino acid, right? The structure of amino acids contains of an amino group. So we have amino group plus a carboxyl group plus a carbon and a R group. Now, what does that look like? First, we have a carbon, right? This is known as an alpha carbon, which is then bonded to a hydrogen. And then we have the carboxyl group, which is C double bond O, OH. Then we have an amino group, which is NH2, so NH2. And then we have this R group here, right? What is this R group? This R group is the only group that differentiates the amino acids from one another. So out of the 20 different amino acids, everything here, that this carbon, this amino group, carboxyl group, this hydrogen bond are all the same. The only thing that differentiates is this R group here. The R group here varies from amino acid to amino acid. For example, a cysteine, CYS, may have a CH2 bond to a SH bond, right? Or serine might have a CH2 to an OH bond here in this R group, which causes each and every amino acid to be different. The only thing that's different here is just the R group, not this backbone here. Now let's talk about polypeptide bonds, right? Now, when we talked about carbohydrates, right, we know that carbohydrates form together with a dehydration reaction. We talked about lipids, which form triacylglycerol with dehydration reaction. How do polypeptide bonds work? Well, it's no different, right? Let's look here, right? Let's just say this was thrown at you. Now, at first, this might be a little bit confusing, like, okay, what's going on here? But we can start to understand some stuff. We see that there's a carbon here to an hydrogen. We have an amino group here. We have another carboxyl group. And then we have some random R group here. So we know that this entire thing here is an amino acid. Same thing here. We know this is an amino acid. How would these two amino acids bond together? Oh, wow, look, we have an OH group and a hydroxyl group and a hydrogen here. This can be taken out of the equation. So we can simply just take this out of the equation, connect a bond together, and then just take H2O out, causing there to be a link between this carbon here and this nitrogen here, creating a polypeptide bond. right? It's as simple as that, right? It's the resulting covalent bond through a dehydration reaction with the removal of a water molecule, right? It's as simple as that. So with that being said, let's talk about the final thing in proteins, right? This usually is the most fun part. Now I'm going to draw here an example. Let's just say that we have an artist that was given a commission to draw a person's face, okay? Now, what an artist would do when you normally draw portraiture is you start by creating this type of very simple, rigid, and linear guideline, right? You have something like this. You have linear work that establishes where your ears are going to be, your eye line, right, your mouth line, whatever it is, where the middle of the face is going to be. Then from here, that's the first step, right? The next step is to then start blocking in values. So, blocking in shapes. So you know where the nose goes. Okay, so the nose goes here. The mouth goes somewhere here. The hair, again, keeping things very simple, goes up here. Then you start to specialize now, right? In step number 3, now you start to create facial values for the person, right? You start to put in your eyes, your eyebrows, and you start, drawing in the shapes for the noses, right? You start drawing in the mouth, the ears, right? So really, you start to differentiate this person from the rest of the people, right? Because you need to make this person look recognizable to the person that's actually in the picture. So now you start to draw in certain facial features. For example, this person might have a bigger chin, a bigger nose, smaller eyes, etc. And the fourth step is to put everything together, right? In a well-developed, a complex artwork, right? You start drawing this in here. You start shading the person, right? You start putting in values, certain wrinkles, certain marks, right? And you start completing a whole person's face. It doesn't take an artist to understand how this process works, right? And just like it doesn't take an artist to understand how this process works, it doesn't take a scientist to understand how proteins actually conform. It's just like this example that I showed here. You start from a very simple illustration and you start to make things more complex.